Hey guys, welcome to the CCMB seminar series. Today we're going to have Bing Ren give a presentation. Bing is really one of the pioneers in a lot of genome technologies that we use in genomics. He's been involved in the ENCODE project for quite a while and in the epigenome roadmap project. And a lot of his research was focused on understanding gene regulation and the mechanisms of gene regulation and looking at epigenomics data types. For example, he was looking at enhancers and was one of the first people to make the observation that H3K4 monomethylation marks enhancer regions in the genome. More recently, he's been doing a lot of work looking at the three-dimensional structure of the genome, with a lot of really seminal papers looking at the conservation of the 3D genome across species and looking at topologically associated domains and those sorts of things. And his presentation today, I think we're going to hear a little bit about how gene regulation can occur through that three-dimensional structure and association of enhancers with genes themselves. So we're really looking forward to it. So welcome.
Can you all hear me okay? So thank you, Alan, for the kind invitation and introduction. So I'd like to begin with this slide where we are all very much appreciate that we are in. So this day, sequencing one's genome is now being done millions of people at a time. But the real problem is trying to understand the meaning of it. And we all know that the majority of the genomic sequences are non-protein coding, and understanding such non-coding sequences in terms of their role in human disease and the human biology is going to be a major challenge today. Many of you I know study does GWAS studies, and you end up typically in a region of the genome that corresponds to nothing. And the question is, what does this non-coding variant do in this disease and that? The typical hypothesis we have is that such non-coding sequences are contributing to disease by altering transcriptional regulation. And there are numerous examples that support these ideas. But to do this, to extend this theory to as a general approach, there are some bottlenecks that we need to overcome. One is that we need a pretty comprehensive knowledge of cis-regulatory elements in the genome. We need to determine the intended targetings of those regulatory sequences, and need to study how sequence variants affect the transcriptional potential of the targeting. This sounds very easy, but in reality, making this to work actually is involve actually a lot of data generation, data interpretation, and also functional interrogation study. So today I would like to introduce you some of the work that we are doing in the lab from three different aspects. One is mapping cis-regulatory elements. Second is trying to define their intended target genes experimentally. And third is to interrogate the functional consequence of a sequence if you delete them in high throughput manner. Let's start with mapping cis-regulatory elements. As Alan alluded, ENCODE project, which was conceived over 15 years ago, was intended to identify cis-regulatory elements in our genome. It started with a pilot project where everyone focused on 1% of the genome by applying a variety of different technologies. And that taught us a few things. One is that there are several kinds of cis-regulatory elements. We knew about promoters. We knew enhancers exist. we knew insulator sequences exist, but we did not know how abundant each category of elements exist. And we also did not know how to find them. Sequence of these elements were not sufficiently distinct enough for us to identify them just by looking at the sequence. So we have to develop new strategies to find them. We and others, as part of that pilot project, tried to apply different tools. So one approach was to study DNA's hypersensitive sites. And that quickly turned out to be a very effective tool to identify all cis-regulatory elements in the genome. My lab at the time was using chromatin modification marks along with transcription factor as marks for cis-regulatory elements. And we come across the signatures for enhancers and signatures for promoters, and finding that CTCF as a reasonable mark for mapping potential insulator sequences. And later on, these technologies initially developed using low-tech, low-throughput techniques were applied, combined with high-throughput sequencing techniques. Now we are using this combined ChIP-seq or DNA sequence are now being applied to many, many cell types to review cis-regulatory elements. So the latest count of different kind of elements are kind of shown here. Now we mapped tens of thousands of promoters, nearly millions of enhancers, and thousands of, or tens of thousands of insulator, potential insulation sites. More recently, we applied this to a mouse embryonic development as a way to learn how this strategy can help us delineate developmental programs. So this is a study that we carried out in the last several years as part of the phase 3 of the ENCODE. As shown here, we try to develop a comprehensive understanding of how development program is controlled. We pick 8 developmental stages and 12 different tissue types from each stage. Some tissue can't be obtained from earlier stages and then were not sampled. Others were not easy to obtain from later stages and were not sampled either. But in total, we profiled chromatin states and the chromatin accessibility from each stage in my lab. And in my collaborator's lab, Joe Ecker's lab, DNA's methylation were profiled of each stage and tissue combination. All the data has been released, deposited to ENCODE DCC, where you can just download and perform analysis on your own. Here, I'm just going to give you some summary of what we have learned so far. If you look at these maps, comprehensive maps, you immediately get a sense of dynamic chromatin modification of different genes. First of all, if you look at some tissue specifically expressed genes, you're looking at different histone marks, and they are present in the promoter or enhancer of the gene in different degrees. So this is a NEUR-D2. Promoter is marked by K4 trimethylation. Upstream of it is a strong enhancer marked by K4 monomethylation and K27 acylation. This is a housekeeping gene. You can see the general active marks localizing to the promoter, and K36 trimethylation localized to the gene body. Hox locus is typically silenced in this stage of development in the forebrain, and you only see K27 trimethylation mark. If you examine across different tissues of the of this gene, you can see that this gene is marked by the active chromatin mark only in the forebrain, but not very much in the other tissues, except for some in the hindbrain and neural tube. NKX2 is a heart developmental regulator, you find it in the heart tissues exclusively, but not in any other tissue. You can also get a sense of developmental dynamics by looking at, for each tissue, for this gene in the tissue, for example, your D2 examining in the forebrain, you find that it's likely silent in the 11.5, 12.5, but become activated after 13.5 development. And correspondingly, this repressive mark, P27 trimethylation, is abundant at this gene locus at 11.5, 12.5, and gradually decrease at 13.5 and afterwards. This is myosin heavy chain. This gene is active in the heart, but in a developmentally controlled manner, especially myosin 6. It's active in the P0. In early embryonic stage, the heavy chain, myosin heavy chain number 7 is active. So there is a developmental switch between the 7 to 6 upon birth. Where are we? Okay, so here. So there is a tool called Chroma-HMM that you can use to identify the combination patterns of histone modification and define chromatin states, developed mainly by Manalis Callis about 5 years ago. So we apply this tool to every stage and every tissue and come out with a total of 15 states of chromatin that can be used as a universal vocabulary to describe all the chromatin modification data we have. So these states, 3 of them correspond to promoters, active, weak, and bivalent, and 5 of them correspond to enhancers, including strong, enhancer distal, in TSS distal, promoter proximal, and weak were poised enhancers, or transcription states, or repressive states, and non-signal. And the distribution of each state are shown here. As I mentioned, the TSS states are localized to the transcription start sites. The enhancers are distributed diffusively along the genome, and the transcription state are in the gene body, a little bit of the transcriptional termination site, and the repressive state, the K27, are slightly enriched at the promoters, while the K9 trimethyl are more diffused. So if you use this vocabulary to look at, to paint the genome, you can now see a very strong dynamic chromatin state in a more comprehensive manner. So look at GATA4. Kristen must be interested in this gene. You look at the GATA4 locus, it lit up literally in the heart throughout the development, and it's become silent in virtually every other tissue except for a little bit in this intestine, and I forgot what this T stands for. This is another gene, GAT1, typically active in the central nervous system, and you find it is to be turned on in a developmentally dynamic fraction in the forebrain, midbrain, hindbrain, and neurotube, but not in any other tissues. So in this pink color indicate polycomb repressive state. So you can see that both genes are silenced using polycomb repressive as a mechanism in any other tissue. So this kind of map immediately can help the developmental biologists to learn or come up with hypotheses about their favorite genes, how they're turned on and turned off. So in the case of GATA4, there are 2 elements that are annotated as proximal promoter or promoter-like, and then a long stretch of sequences marked as yellow as enhancer or strong enhancers. And then there is a region here that corresponds to transcribed regions. So you can basically guess that this gene is transcribed from at least 2 different promoters and with multiple clusters of enhancers driving its expression. K27 has been associated with transcriptional activation. So by looking at where they are, what we are trying to see is where determine local enhancers. For example, now we can take these regions marked by K27 peaks and ask, do they correspond to tissue-specific enhancers? So my collaborator, Len Panaccio at Lawrence Berkeley National Laboratory, performed transgenic assays by taking these predicted enhancers tethered with a reporter gene and injecting into the embryo, wait until the development stage 11.5, then perform the staining to look at reporter expression. So in nearly every cases when you have such a strong enhancer, they would direct expression of that reporter gene in the tissue where you detect such enhancer activities. For example, limb. This is the limb enhancer, and we predicted it would be in the limb, but the reporter shows that it's active in the limb. This is elements that are showing activities in hindbrain, midbrain, neural tube, and indeed, when you perform reporter assays, this is the patterns that corresponding to these regions. And this is a forebrain specific element, and indeed, the staining of that reporter shows forebrain specific activities. And this is a heart enhancer, and it's showing up in heart. We've done this for several hundred predicted enhancers. And those predicted enhancers are ranked into several categories. The top 1,500, the mid-tier from 15,000 to, top 1,500, from 1,500 to 3,000, and the below 3,000 based on the K27 oscillation levels. So the top tier appear to validate much more likely with nearly 60% of the elements driving reporter in the right tissue, and with nearly 80% of them driving a reporter in some tissues, not necessarily in the right tissue. If you go down below 1,500 to 3,000 or lower, the validation rate drop considerably, indicating that the levels of K27, the stronger, the more likely this is a very active enhancer. Once you drop to lower levels, the chance that it serves sufficiently high-level enhancer also decrease. So this type of information would be very useful for biologists to interpret these maps. When you see a very strong K27 oscillation map, you can put that into perspective, how likely it's going to be validated using this transgenic mice assay. We also perform chromatin accessibility assays using the attack-seq approach. I won't go into details about this method. It was developed by William Greenleaf in Stanford a few years ago. It's a very quick and easy way to look at chromatin accessibility. So we apply this to the same tissues and stages, except for the 10.5, and attained nearly 720,000 elements in any one tissues. And this is a comparison of those elements to the human genome, with roughly 70% of those elements that we identified showing conserved sequence in human. And of those conserved sequences, we found roughly, so out of this total, 40% are also identified as candidate cis-regulatory elements in the human by DNA hypersensitive assays. But then we discovered also nearly 30% or 200,000 elements that were identified as potential cis-regulatory elements. Sequences are conserved, but were not previously identified in the human genome as hypersensitive sites. So you can look at this as potential new functional elements that were defined using this assay, because we're looking at the developmental stage that were not previously studied in human. Or you can look at this noise, but I think the fact that the sequence conservation is high indicates that a lot of them, if not the majority of them, are likely functional elements. And then we ask, are these potentially conserved cis-regulatory elements enriched for GWAS hits? The answer is absolutely yes. This is a table showing different tissue and the developmental stage combination. So the color, the redder the color, the higher the enrichment for specific traits, such as in the top here, these are enriched in forebrain, midbrain, and hindbrain, so nervous system related enhancers. And the type of traits that we found to be enriched in this category are also related to nervous systems, such as schizophrenia, educational attainment, and down below here, electrodermal activity. So attention deficit hyperactivity disorder, for example. And bottom here, these are elements highly enriched. These are elements in the liver. And if you look at the kind of disease that GWAS hits are enriched in this kind of liver-specific enhancers, you are finding a lot of metabolic pathway traits, cholesterol, triglyceride, for example, that are enriched. So what this suggests is that a lot of the symptoms that we study in humans, they probably have their developmental origin in terms of as a root of their initial disease pathogenesis. So all the data are being released, have been released to the ENCODE BCC portal. You can download from this website, and essentially this is what you see when you first log into it. They give you a very convenient view of assays and samples, organisms, and this is a table that you can basically click and download all the data. Now, as I mentioned, through this type of effort, there have been millions of cis-regulatory elements identified literally in the human and now in the mouse genome. These elements are showing strong enrichment for sequence variants linked to human traits and disease. And this support further the hypothesis that non-coding variants contribute to disease through regulation of gene expression. Now, the question follows is that how do we move to the next step, which is to find what genes they control? And this we take advantage of, well, first of all, why is this question relevant? It's because a lot of examples have been reported that the gene and its enhancers can be far apart, sometimes as far as a million base pairs, and certainly have many genes in between. So it is not safe to always assume that an enhancer activates the gene next door. But the question is, how does an enhancer element act over a distance? It's now well accepted that DNA actually can fold and allowing the enhancer to loop back to the target genes through spatial proximity, and therefore they can control a gene even though the linear distance is far. Can we take advantage of this looping between enhancer and promoters to identify target genes of enhancers? And that requires us to understand how spatial organization of the genome is arranged in the human cells. We have Joop Dekker and Res Lieberman to thank for developing a powerful tool called chromosome conformation capture, also known as Hi-C. This strategy requires fixation of the cells with formaldehyde and then digestion followed by ligation in a so-called proximity ligation reaction to allow two sequences that are close to each other to be ligated together and isolated. And then this can be sequences in mass to give you a genomic view of chromosome interactions. Each dot here represents a frequency between two sequence elements. Zooming in and turning this 45-degree, you will see this patterns of local chromatin interactions with this regions showing very high intensity of interactions, followed by another domain, and they are separated by a short boundary. And we call this, topologically associating domains because they are defined by the spatial interaction of the genome. It turns out that such domains provide a very nice framework for us to understand how transcription is coordinated in a genome. This example shows that the boundaries are essential for proper gene expression patterns during development. If you delete boundaries, for example, in this locus, what you end up is a developmental defect, such as brachydactyly, resulting from ectopic activation of developmental regulators such as PAX3 due to the deletion of the boundary. So such examples illustrate that maintaining the chromatin organization is crucial for proper gene regulation. In terms of mechanism, such domains are considered to form due to a pair of players, CDCF and cohesin. It's because the boundaries of TAD, topologically associating domains, are enriched for this transcription factor, CDCF, and also the CDCF binding sites at the boundaries tend to adopt convergent orientation, making them essentially very possible to form this spatial arrangement. Cohesin is a ring-shaped molecule that could wrap around this two-strand of DNA. And cohesin also is known to interact with CDCF. So according to one of this model, this cohesin can traverse the genome in this opposite direction, and they will stop at the CDCF binding sites. While doing that, the intervening sequences are extruded out and forming its own domains. So that's how genome is arranged, and we know now that such topologically associating domains are very stable during cell divisions, and it's the same cell type always have the same type of domains. Even different cell types can have a largely overlapping set of TADs, indicating that they are a very stable structure throughout development. Within TADs, there are loops that you can identify. For example, if you use high resolution analysis by sequence the maps deeply, you can actually see regions that are having local high frequency of interactions that are separated by regions that are not as highly interactive. This, we call them loops. And finding loops typically, or in theory, should give us idea about how enhancer and promoters are interacting. One approach that we have recently adopted was to use capture approach to enrich for promoters from the Hi-C library and sequence only promoter-centered Hi-C interactions. So this type of data requires a bit more computational effort. So when we get the data, it's almost like a Hi-C. Imagine this Hi-C type of map, but it's highly biased towards the promoter regions. So we have to remove experimental biases due to fragment length, due to the probe capture efficiency, and also consider the distance-dependent signals. So when we and then we calculate a probability value whether the interactions, the observed frequency of interactions are unusual. So when we do that, we can identify many promoter-centered interactions in the fibroblast cells, as shown here. And when this is compared to the high-resolution Hi-C data, you can see that in large part, these two are quite similar. In fact, this is a statistics, or the similarity between promoter capture Hi-C versus Hi-C data is very similar compared to random controls. So this gives us confidence that using the promoter capture Hi-C, we obtain essentially same information for each promoter compared to a whole genome Hi-C, but at only a fraction of the cost, roughly 10%. So this gives us the power to analyze dozens of tissues, obtaining enhancer-promoter interaction maps at the cost that we can bear. So we apply this to 27 cells and tissue types for which we previously analyzed as part of the roadmap epigenome effort, so that now we have both epigenome maps and promoter-centered interaction map for the same cell type, same tissues. And altogether, we're finding about nearly 300,000 enhancer-promoter interaction pairs at a resolution about 5 kilobase. The median interaction distance between any pairs is 140 kilobase. This pie chart shows that out of this 300,000 pairs of interactions, almost 7% correspond to promoter-promoter interaction. 36% correspond to, 56%. 37% correspond to promoter-enhancer-like interactions, and then nearly half correspond to interactions between a promoter and the regions that not yet marked in that tissue. So there's potentially a lot of sequences that may play a role in the genome, but not yet identified by chromatin-based analysis. How? This is just cis within the local chromatin. How reliable is this data? Let me show you a couple of examples. We use eQTLs as a expression quantitative trait as a ground truth. So we look at one region, for example, VL, DLR, by eQTL, it has a sequence about a couple hundred kilobases away that are strongly associated with the expression variations of this gene in the heart. And by looping analysis, we found that this locus is specifically looped to the promoter of ELR. So there's another region here that has a slight local eQTL enrichment, and then there's another loop that we identified were not supported by eQTL analysis. When we compare this in a more genome-wide fashion, we're finding a tremendous amount of overlap between eQTL and the loops that we identified in the corresponding tissues. And by random control, you'll find much less overlap, much fewer overlap. So this indicated that there's a strong signal that were captured by the looping assays. This is in 2 other different tissue types, the same type of eQTL analysis as data generated from the GTAX consortium shows strong overlap with our capture promoter enhancer interaction. This is another view of the data showing that in these this gene that are typically expressed in the hippocampus, there is a strong promoter enhancer interactions that are detected in the hippocampus, but not in most other tissues, suggesting that tissue-specific interactions in this case correspond to tissue-specific expression. So this map, obviously, in theory, should give us indication of target genes of potential GWAS hit. Indeed, we examined some of the known GWAS hits that have been well studied. In this case, it's FTO locus. It's being shown that FTO locus actually that correlate with the obesity is controlling 2 genes, IRX3 and IRX5. And in our case, we found very strong looping interactions between this locus and both genes in multiple tissues. So this gives us hints that what we are finding are indeed potentially useful. Another indication of the reliability of this is focusing on this unexpected set of interactions that involve 2 promoters. 7% of those potentially correspond to a pair of genes where the promoters are spatially close. The question is, are these real or are these just due to our experimental artifact? Again, we first examine whether the promoters that are interacting with each other are showing activities that are correlated across the same set of tissues. And indeed, you can see that there is a very strong Pearson correlation of different quantum marks between promoters that show spatial interactions compared to random pairs of promoters. This suggests that indeed there are potential correlations between this transcriptional correlation between the genes or promoters that are interacting in space. Second evidence is, again, showing by eQTL analysis. Here is a gene that BL-cap that shows strong promoter interactions with another locus, GHRH. And now, if you look at the eQTL data, this region include the GHRH is actually a strong eQTL corresponding to the BL-cap activities. So when you compare all the promoter-promoter pairs that we identified by promoter captured high C with eQTLs, there is a very strong overlap compared to random expectation. So what this means is that we are indeed finding true signals using the promoter capture assays that can be verified using eQTL. So this map, how do we use it to study human disease and traits? One way we do this is by first categorizing all the traits over 907 categories into groups that share a similar set of genes based upon the SNPs. So this is a heat map showing those 30 groups of traits. With each group, the SNPs identified by the GWAS studies have the target genes that are overlapping strongly with each other, with this Pearson correlation showing up here very strongly compared to random. So this what this gives you is, if you look at, so here, cluster 1 are traits that are named here: Graves' disease, Batchet disease, psoriatic arthritis. These appear to relate to inflammation. The second category that share very similar genes that using these maps are Parkinson's disease, ADHD, Alzheimer's disease, frontotemporal dementia, for example. These are all related to central nervous system. So why is this better than just using nearest gene as a predictor? This is how, if you use the nearest gene, the p-value that you show such enrichment is not as strong as if you use the putative target genes. So this is for cluster 1. If you look for functional enrichment of functional categories, the top category is peripheral blood leukocyte, very strongly enriched. But if you use just the nearby genes, it's less strongly. The second category, systematic lupus erythematosus, this also is more significant if you just use the nearest gene to conduct this analysis. So what this tells us is that if you use this map, you're more likely to uncover the true biological pathways than just use the next gene neighbor. So using this map, you could now analyze pathways involving pathogenesis of different traits. So the category 1, 17, 21, and 29 shown here appear to share similar pathways that are enriched in their GWAS SNPs, and C4 and C14. They also share similar pathways that are involving blood coagulation, platelet activation, and so on. So the proposal is to use these maps to really identify common shared pathways among different diseases and use that information to help us devise strategies to treat different diseases. That's obviously going to be a lot of work ahead of us. In summary, this line of work will give us candidate targeting for potential causal genetic variants and help us understand the molecular phenotypes from the GWAS study. Obviously, many questions remain, and I won't be able to address them in the near term. But one of the questions that we try to study is provide functional data for elements that we identified using this high throughput approaches. And here I'd like to introduce a method called CREST-Seq, which stands for Cis-Regulatory Element Scan by tiled deletion, and sequencing. This involves using a pairs of guide RNAs expressed in single cells in parallel to create deletions. And we can generate thousands or tens of thousands of such different pairs, such that each pair would delete a overlapping sequence in the genome. For example, we pick this locus, we can design the deletions, and they are basically covering this region in 20-fold coverage. The strategy involves you make such deletion design, generate lentiviral vectors that express 2 guide RNAs, delete and introduce double-strand break with 1 or 2 kilobase apart. You introduce this lentivirus into cells that are in a culture in a 0.1 multiplicity of infection, so that each cell receives no more than 1 lentiviral particle. And after selection for those transfected or infected cells, you sort based on the expression of the target gene. In this case, the target gene that we select for validation of this strategy is OCT4, where one of the 2 alleles has been tagged with GFP, so that we can use GFP expression to monitor the expression of our OCT4 in that allele. In this experimental system, we focus on human embryonic stem cells with this tagged OCT4 and designed 11,570 pairs of guide RNAs to generate, on average, 2 kilobase long deletions. Every pairs of deletion that are next to each other overlap by 1,800 base pairs. So they have, 1,900 base pairs. So that's roughly 100 base pair step sizes and the 20-fold coverage. This design allows us to interrogate each element multiple times for its function, and the step size gave us the resolution whether the element is active or not. To screen for this, we infected the cells with the lentiviral vector, grow the cells, and then screen for those that have lower GFP expression, but maintain regular levels of the OCT4 so that we ensure that the cells are still pluripotent because the other copy of OCT4 is still expressing a normal level. So this is the cells that we isolate. So they are no longer express GFP, but the other copy of OCT4 is still functional as a control. If you look at cells without infection, very little cells meet these criteria. We also identify high GFP population as another control. So this, when we sequence the insert of the lentiviral vector to count how many times a particular guide RNA were enriched in the GFP low population versus the control total population. And that is plotted using this MA plot, where the mean read count is on the x-axis and the y-axis showing the fold enrichment. The ones that labeled red are those that are considered significantly enriched in the GFP low population compared to this control, and there are about 500 of them. This includes all the primers, all the guide RNA that are designed against the GFP gene itself, shown here as the green circle. As a negative control, we designed guide RNA, the same sequence, but lack the PAM sequence so that they don't target specific DNA, and they're not showing strong enrichment. Only 2 out of 500 were showing up, so indicating a low false negative rate. Another control, if you look at the high GFP population, you see this, this red population now shows depletion in this high GFP. This is absolutely consistent with them being required for GFP expression. And if you knock them out, you won't see them to be highly represented in the high GFP cell population. We can, this is a map to show all the designed guide RNA pairs, and this is their abundance, how many times they were plotted. Sorry, this is the ones that we show as high enrichment. And this is the OCTA4 locus. And as is expected, OCTA4 locus, there are many deletions that would affect its expression. But then there are many other locuses that shows pile up of this guide RNA pairs. To identify systematically regions that have functional importance for OCTA4 expression, we designed a the following test. We use first negative binomial to rank all the guide RNA pairs from top to bottom based on their enrichment levels. And then we identify those genomic regions where multiples of such guide RNA pairs are found to be highly enriched. And this, we use a robust rank aggregation test to show essentially whether there's a strong bias for overlapping guide RNA pair deletions are in the top of the rank. And that p-value can be plotted, and basically you can run through every 100 base pair and plot all the p-values. And regions that show such enrichment, basically strong bias, are plotted here as high, and this region that don't show any enrichment of guide RNA pairs are obviously no signal. So you can basically, so basically the original map of guide RNA pairs now can be added, annotated using this signal map, and showing that this region have many guide RNA mediated deletions that has very strong scores. There's another region that shows a score here and here and here. This region high up here have a high density of such functional regions as identified by the CREST-Seq. And if you zoom in, you can find that many of those peaks are actually regions bound by transcription factors in the human ESLs, or showing association to active chromatin marks, as we have expected these regions to be. This is a summary of all the regions scored as positive. A total of 45 regions were scored as positive by this assay, meaning that they are necessary for optimal OCTA4 expression. And their overlap with active chromatin marks are shown here. So altogether, all but 5 of them are associated with active chromatin marks or transcription factors or PAL2. These are non-marked regions, a small minority, but still a significant minority, indicating that this assay is capable of uncovering regions that are not annotated by biochemical signatures. Another region that to pay attention is that nearly 40% of this positive elements correspond to promoters. This came as a big surprise because we typically think promoters only care about their local neighbors. They don't worry about anything else. But apparently, these different gene promoters, when you delete them, can contribute to reduced OCTA4 expression. So we want to do validation to show that these are really true positives, not false positives. And to do that, we designed the following assay. We design a pair of guide RNA, generate clones of cells where the deletion occurred in one of the two alleles or both alleles. And here's the phenotype you would expect. If you delete the enhancer from both alleles, GFP obviously will be low. If you delete the enhancer from one allele that corresponds to the non-tagged version, you won't see a reduction of GFP. If you delete this enhancer from the tagged version, you will see a reduction of GFP. So, so we selected 6 elements for validation using this approach, and they're marked here. In each and every case, we see a reduction of GFP expression when we delete the element in cis, but not in trans. The cis element deletion is shown in the yellow, and the trans element deletion is shown in red. And you can obviously see in every case, the green one is lower and shifted throughout the lower GFP side compared to the red ones. This is a FACS plot showing individual cells GFP expression. We did this in 2 time points. One is 25 days after we did this experiment, isolate individual clones, and do the FACS sorting. Another is 50 days. And interestingly, 1 out of 6, we show a reversal of the phenotype where the cis deletion no longer show a reduction of the GFP. But in every other case, the reduction is robust and persists. So we name this kind of element as temporal enhancers, meaning that deleting such enhancers have a temporal phenotype. This region corresponds to one of those unmarked region that we detected by this assay, but they were not marked by any. It was not marked by any of the active chromatin mark or transcription factor binding, and you can see that deleting these sequences result in the robust reduction of OCT4 expression. 3 of these tested elements correspond to promoters. So we initially deleted a 1 kb segment of promoter. And the reviewers asked, oh, that may include proximal promoter regions. So we then delete the core promoter of 200 base pair region in 2 different ways, so that our deletion is not causing off-target effect. And in each and every clones we isolated, the cis deletion always resulted in a reduction of GFP compared to the trans deletion, indicating that promoters indeed are, in this case, required for optimal GFP, OCT4 expression. This is not due to the product of those genes, because when we did RNAi, we can reduce the transcript of each gene and protein levels of each gene, but without reduction of OCT4 expression. So this is likely because the element is required, not the gene product is required. So how could this be possible? I think one hint came from analysis of a 3-dimensional organization. So this plot shows the average ligation frequency between OCT4 and each of those promoters that show positive CREST-Seq signals. And the gray shows the genome average for between promoters and its neighbors. And what you can see clearly is that out of those 17 promoters that show a positive, basically requirement for OCT4 expression, all but 3 are highly interacting with OCT4 in terms of the spatial distance. Another way to look at this is when you try to predict which promoter out of nearly 300 genes in this locus are OCT4 dependent, are regulating OCT4. So we design a random forest predictor by incorporating data such as high C and the transcription factor binding and active chromatin marks. And when we do this, we can predict the which promoter to be active very actively, roughly with ROC of 0.9. So, and then we drop off each feature individually, ask which ones are more important, are required for the accuracy. And the first and rank number 1 is high C. It's by far the largest single predictor of promoter being required for OCT4. What this suggests is that spatial interactions between the promoters of OCT4 and promoter of other genes are a defining feature of this promoter required for activation. So what these functional studies taught us is that we can now design an unbiased strategy to identify enhancers in the genome, and show that actually a lot of promoters can act over a distance to control genes that are not even related to the function. This echoes back to the promoter capture studies, where we see a lot of promoter interactions coming together. We argue that a lot of transcriptional control in the genome is controlled by such promoter and promoter interactions. So I know I'm kind of exceeding time. So in acknowledgement, I like to emphasize several key players. So the first ENCODE study I presented were led by David Gorking and one of your own product, Yanxiao Zhang, and Yuan Zhao, a graduate student, and 2 very hardworking research associates. The single-cell attack-seq, which I did not have time to go through, is listed here, and chromatin interactions was done by Anthony Schmidt and a postdoctoral fellow in Kyung, who has now have his own lab in Korea. And CREST-Seq was invented by Yare Diao, Rongxin Fang, and Bing Li. And we have our collaborator to thank, and also funding agency here. Thank you for your attention. Happy to answer any questions if I have gone through too quickly. Yes, back over there. We haven't investigated that question directly, but there is evidence that that requirement is not there. Let me show you this example. Here, DL cap is actually not expressed, but sorry, I think but this is the interaction that shows up in IOTA. Sorry, not DL cap. DL cap is expressed here. It's from the negative strand, but GHRH is not expressed, and yet it shows very strong interaction. So it's possible, but not definitive in this case, that the promoter of the GHRH may be driving DL cap expression. But I take this, I mean, there is a caveat. So of the 17 promoters that we identified that are required for optimal OCT4 expression, there is a strong correlation between their activity in terms of transcription levels with their being required for OCT4 expression. So I would say there is a strong preference for genes to be turned on, for them to be also used as regulatory elements that are spatially close, but not absolute, I would say. Yes.
So are they being shared at like transcription factories? Are they localizing? Have you visualized them by like maybe DNA fish? Are they co-localizing at certain factories together? And this is why you see these interactions?
Yeah, I think that's one of my models to explain this. I think spatial interactions may be consistent with them being from the same transcription RNA polymerase factory. We haven't been to establish techniques to visualize them in live cells. It's really hard, but I think that's a very plausible explanation for what we observe. Back over there. Yes.
So the first question is do you think this distal regulation and the proximal regulation are mutually exclusive?
No, but there is I think they're mutually exclusive, meaning that they can occur at the same time. But in each cell, there's always a dynamic. So the enhancer may interact with this promoter at maybe first second, and then the next enhancer may come in and kick it off for the next second. So yes, if you look at the duration of time, they can co-occur.
So the follow-up question on this is now if you put this into perspective, when you do the gene deletion, actually you're changing the RNA structures. So how does that come into play when you try to explain this kind of distal regulations?
Yeah. So to rule out the gene product as playing a role, we use RNAi to reduce the gene product, and we did not observe a downregulation of OCT4. So that's one piece of evidence we have against that model. Yes.
Really very nice talk. I'm kind of curious for the OCT4 example, when you deleted any of the other genes promoters, do you see effects upon the promoters other than of OCT4? So is there?
Yeah, we did look at another gene, TLC, and we did not see that being affected. So the HLA-C is another gene that is about 100 kilobase away. Even though some of these promoters are hundreds, 500 or 900 kilobase away, they're not affecting the HLA-C gene.
So in one case, you say there is a tempo enhancer. Do you study this in cell culture, right? What can you explain? I mean, what's the mechanism? How would a temp enhancer would work?
Yeah. Temp enhancer was found, yes, in this system. How does it work? Do I have a picture? No. So there have been always suspicion that each individual enhancer probably can be compensated by other enhancers in the same region. So deleting single enhancer are not expected to have an effect. And I think in our case, we were fairly surprised actually deleting any of those 45 regions did lead to an effect, right? But in the temp enhancer, what I suspect happening is that the effect is compensated by other enhancers after extended period of culturing. But obviously for some other enhancer, when we delete, such effect was not compensated. So we are not very sure what's the difference between these two categories of elements, that one is compensated and other is not. All right, thank you. Thank you very much. Oh, it's my great pleasure.